Great. All right, so, okay, this talk is about estimating dark ocean respiration through the ETS activity. And I want to start just with a very simple slide. This is this one, just stating that we have two waves of addressing the biological pump in the ocean. It's like the two sides of the coin. One is looking at the carbon fluxes in the water column and how they are mm, just how they decrease and how they attenuate and so on. We're just looking at these carbon fluxes. And the other one is looking at the remineralization of these carbon fluxes. So in, in a simple way, I would say that it's more straightforward to look at the remineralization, because when you look at the carbon fluxes in the water column, and I don't have to say this, and we are not going to discuss this today, but it's so complex, and many of you are working on this. Every of these single fluxes is so complex from the methodological point of view that is hard to understand how we can put all these fluxes together and reach a consensus about it. Just looking, for instance, as the sedimentation of particles, we all know that sediment traps, particularly in some regions where there are lateral affection, they underestimate these carbon fluxes. We know that the new vision cameras not always match with what you collect with the sediment traps. We know very little, for instance, about how much DOC is absorbed in these particles, and they are sink down in the water column. And we have always lived with the idea that these particles contribute largely, or is the main flux of carbon to the deep ocean, but in some regions probably they are not. And then we have, for instance, the flux mediated by the vertical migrants that could be circa daily or circa diary, or can be seasonal. They could be fishes or could be crustaceans, that they could live in several layers in the ocean. We still don't understand very well how is this later migration strategy there. And looking at the active flux is also a nightmare, because the animals, they feed up on the surface, and then when they are migrating down, they are respiring, they are defecating, they are excreting, they are predated by others. So it's so difficult to quantify this. But even the most simple fluxes are even difficult also to quantify them. And then what to say about the physical processes? So we know that there are a bunch of physical processes going from deep mixing, like convective overturning circulation in the water column. I think that's more simple to try to quantify how much carbon is exported by overturning circulation, but there are many other processes at small scales and at very small scale, like there's a mesoscale, that they definitely play a key role in transporting suspended organic particles, but also this organic carbon to the deep ocean. So there is a lot of knowledge, but there is also a lot of unknowledge about all these fluxes here. So one would say, and obviously, what about lateral advection? If we are in an area close to the continental boundary, but you have a lot of carbon exported from the coast to the open ocean, and we don't know exactly how much this carbon plays what's the role that they place in the carbon cycle. So it's really a very complex way of looking at this. I don't going to discover anything about this. I just want to pose that this is, you probably all agree, it's a very big challenge to quantify all these fluxes separately and try to balance them with a respiration rate. So, in simple terms, should be easier to look at respiration, because it's a much more straightforward measurement. If you look from the biochemical point of view, you can look at all the bull respiration in the water column. If you want to look at more ecological processes, you can look at microbial zooplankton respiration and so on, but should be more straightforward, but they are not. It's not. And that's the reason, because we have this SCORE working group here. It's really complex to measure respiration in the deep ocean or in the dark ocean. So let us start just with just a very brief overview of biochemical approaches. We are going to talk in other talks and other presentation about this. Jack and Pepe, I know that they have like some presentation about that, but just saying that these, to me, they should be the gold standard of the method, all these biochemical approaches, basically because they integrate these long-term and large-scale processes and avoid sample enclosure and manipulation. And that's the main reason of this. Obviously, there are some cons like uncertainty in the fluxes, there are the mixing effects, uncertainty in the estimations of eddy diffusivity, both in the vertical and in the horizontal, and also all the models need some independent verification here. If we go to the ecological approaches, and there are probably a few more than I have outlined here, but there are three main approaches here. One is to look at the in vitro oxygen consumption, that we can do it through bottle incubations or respiration chambers. The prokaryotic growth efficiency. Prokaryotic growth efficiency is like what we say in Spain, is like a fish biting its own tail. I think in England you say like a dog chasing the tail or a snake biting its own tail. It's like that. I mean, if you have the prokaryotic growth efficiency of the prokaryotes in the ocean, you can just measure in prokaryotic production, which is pretty easy. You can go and derive or infer respiration. But to have the prokaryotic growth efficiency, you need respiration. So most of these prokaryotic growth efficiency are in some way artificial and are based on a few results that have been measured in the water column, or most in the surface waters, or most they are derived in a very, let's say, a conceptual way, like we would expect to have in the deep ocean. And the third approach is the enzymatic measurement that we will be talking about in this presentation. So all these approaches, they have some pros, they are precise in some way, and mostly they differentiate ecological processes, which is quite interesting and important. But there are many cons. They only address short-term spatial and temporal variability, and when you want to extrapolate this to the larger scale and to the long-term scale also, is really difficult. They rely on conversion factors, many of them. Even measuring primary production prokaryotic production relies on conversion factors. And also, and this is one of the most critical one, is the sample manipulation, bottle effects, and Gerhard was talking the other day about the pressure effects also when you collect samples. So going from the in vitro oxygen consumption, we have three ways of looking at this. One is with the microwinkler in incubation bottles. Microwinkler is a very sensitive method. You can go pretty down to very low rates if you were very careful with that. But they are always affected by pressure effects on microorganisms and bottle effects, because you need to incubate for doing long time, more than 2 days, and there are changes in the prokaryotic assemblages, and there are changes in the balance and in the activity of the organism. So there is a change in the community that is growing during those 2-3 days inside the bottle. So you need to keep for more than 2 days their incubation there. And also you have all these pressure effects on microorganisms that Gerhard and Christian have been reporting in the last years about that. The other option is the mass spectrometry, which is very sensitive also. It's like the microwinkler, but and it has an advantage over the microwinkler, is that you can incubate in vials and you can have like time series. It's not like an output sensor, but you can have like time series continuously. But if you have worked with this, it's like a nightmare, at least to bring on board a ship, because you need to maintain very constant temperature, and it's difficult to work with this equipment there. And then we have this new generation of high precision oxygen sensor, the Optos, which probably is the future. And as far as I know, but probably I'm not right, the Optos still, they don't have the same sensitivity as the microwinkler or the mass spectrometry, but they are going in the right direction. And the good thing of the Optos is that you can include them in these chambers, and you can record in time series and have slopes of oxygen consumption and so on. So from this, let's say in vitro oxygen consumption, I think this is the future, the Optos in incubation chambers. And I hope that either Dominic or Matthew, they were giving us a talk about this and illuminated us a little bit on how this is progressing in the field. So up to now, there are very, very few published studies on respiration in depressurized samples. And here are 3 examples. I don't know many more. And these are not comparable, to be honest. One is the very old paper of Vidand and Benner. These were, if you look at the depth where they measured, was 100, 500 meters. It was in the Gulf of Mexico, extremely highly trophic area. So Ron Benner, he told me personally that their data was not comparable to anything else. So it cannot be comparable to any mesothelial respiration rate that you would expect here. Then there is the work that we carry out in the Northeast Atlantic near the Canary region in 2005. And this is the mesopelagic layer, clearly. And this is a region which is highly affected by advection of organic material. So it's a very, very productive region in some way in terms of bacterial activity. There were many proxies of bacterial activity, not only respiration, but other proxies that give you the idea that the bacteria were really active in this region. And I will tell you how we measure respiration now. And there's also this paper led by Thomas Reinhaler with Gerd Hendel, where also Carl Robinson and myself collaborated here, where respiration, they measure respiration in the North Atlantic down to the bottom, down to 3,500 meters. The respiration rates were also pretty high here also. So we will see how this converse later on respiration ETS ratios. So for the study of this study of the one we did in the Northeast Atlantic, what we did is we realized that when you enclose the community in the bottle, they were growing in 2-3 days. So we monitor this growing of the bacteria here, and we measure both prokaryotic production and prokaryotic biomass here, which parallel the consumption of oxygen in experiments here. So we estimate prokaryotic production by 3 different approaches, or 2 different approaches. One is a change in biomass, but also looking at the average leucine and thymidine uptake here. And we also estimate the average prokaryotic abundance. I think we use this relationship to backscale later on. So I realize this is not perfect, but this is probably the least worst way of estimating respiration, where you have increase in or changes in your community inside the bottle during the incubation. So what we did is a backscaling here using this regression or this correlation here, saying, well, if we have in situ this amount of bacteria, we will have the respiration. So so in fact, the respiration rates well about a third of what we measured just at the end of the experiment, just looking plain or straight at the end of the experiment. And then we have the electron transport system method. And I wanted, in honor of Ted Packard, I wanted to bring pieces of his first manuscript that he published in Journal of Marine Research in 1971, where he developed the ETS technique, which measures potential respiration by detecting the respiratory electron transfer activity in all these the hydrogenases, and cytochromes that control respiration. Collectively, all these enzymes are known as the respiratory electron transport system. So, and this is quite important. Any enzymatic assay needs to be measured at its maximal level of activity. And I say this because we will have this discussion later on this group, which is really interesting because there have been other approaches with zero substrates, which not adding any saturating levels of substrates. And there are some differences between one of the other methods. And so this is the classical kinetic enzymatic study where you saturate with these protein substrates with NDH and NADPH in this case. In the beginning, it was also succinate, but it has been removed in the last versions of the method. And then you use an electron acceptor, which is the INT, which replaces the oxygen here, and is reduced to INT formescent. So when it is reduced, it produces red color, and this red color is what you measure in your spectrophotometer. So it's a very, very simple assay, in fact, and it's very also very easy, very straightforward to apply. In the last years, we have to say there have been some changes in the site in terms of reducing the amount of water that you have to filter. I remember when I started filtering this, I filtered like more than 20 liters of water. At this depth now we are filtering in the deep ocean like 8 liters. So you can concentrate your samples. You can also do like continuous kinetic measurements. So there are some variations that have improved the methodology, but basically is the same. And this method basically it says measure potential respiration, and we need to be very clear on that because measure the respiration that would be under saturating levels of substrates. It's a fast, it's a sensitivity method, and it allows collecting a large number of samples. So you can increase your resolution both in the horizontal space, just looking at any variability like mesoscale and mesoscale, frontal variability, anything, but also in depth. So you can look in depth down to the deep ocean. And this is quite interesting. You want to look at like layers of different respiration rates in the ocean. And it basically avoids potential and pressure effects. But the weak point of any enzymatic method is that you need to convert your potential rates to your actual rates. And this conversion is not, or this ratio for the conversion is not a single ratio. It's not so straightforward. It varies. And it varies according to many, probably, or several issues like the metabolic state of the organism, the substrate that is available to the organism, not only the amount or the quantity, but also the quality of the substrate. Perhaps also other environmental parameters like oxygen, like temperature. We know very little about this. And these are critical to try to use them if we want to parameterize the relationship between respiration and environmental parameters to implement, for instance, biochemical models. We have very little information of the relationship between R and ETS in the dark ocean, but we have some information in the surface ocean, in the epipelagic ocean. And I wanted to bring you up here one of the, I think this is the more complete database on respiration and ETS in the surface ocean. All these gray dots here belong to this paper we published in 1995. They belong to different cruises from different oceans, from the Gulf of California to the North Atlantic, to the Southern Ocean, to the Mediterranean, to the Canary Islands, to many places, in fact. And it was a pretty strong and robust correlation between the respiration and ETS. And here superimposing these in colors are some more data from a mesocosmos experiment that we ran not many years ago, looking at the influence of CO2 and nutrient fertilization on, in this case, on the respiration and ETS activities. All of them align the same line. You can say, wow, we have a very robust relationship here. But the truth is that there's a lot of variability, intrinsic variability in between them. So there were variability. You go to the former paper where we describe this, you see the variability in the different regions. And there is also variability here in these two phases, for instance, when they were from oligotrophic, mesotrophic to eutrophic conditions here. So we need to understand this variability if we want to apply respiration at local regional scales. And I think that's important. It's not only to try to put everything on the course of a regression line, because always it will be correlation, because it depends on the biomass, basically. So I think we have to search onto this. I mean, this is good to know that they correlate, but we need to go into that and try to explore what is happening. And how we can translate it to the deep ocean is even more complex, because in the surface ocean, you have a change of bacteria and phytoplankton and so on. And we know, for instance, that large respiratory ETS activity and respiration activity is linked mostly to phytoplankton blooms, but then there is a drop when you have bacteria. So we are trying to understand what is the relationship between respiration and the shifting community structure. But in the deep ocean, the rates are so low that this is really a challenge to do it. And what we know from the deep ocean, we know very little. And I want to go back to this paper, which I love it. It's a paper from Christensen and Parker also from 1980, where they culture 5 batch bacteria cultures with different adding an organic substrate during 2 different phases, with the growth phase when they have a lot of substrate, and the senescent phase when the substrate was depleted and there was no organic matter. And you can see that during the growth phase, the RETS was matching completely. So it was almost 1. But with the senescent phase, the respiration dropped down completely, while the ETS, all enzymatic activity, remained pretty constant, at least for a while or for some days. So that means that the RETS dropped down an order of magnitude. So what is happening in the ocean? What is happening in the dark ocean? What is happening in the particles? What is happening in the free bacteria? So what are we really measuring there? In the paper of in the work we did in this one where we backscale the respiration, we got this average value of RETS, which is has a lot of sense if we compare it with the values of Christensen at all, because it's a high value, more close to 1 than 0.1. That means that they were active bacteria growing up, but they were not absolutely active, like it was in the growth phase with a lot of nutrient replenishment of organic material. And then we have also this study by the one from Thomas, where the RETS is pretty high, and is, I mean, just looking at this one, we say, well, this is unrealistic, but we need to think about what make to have this high RETS value at in the deep ocean also. So there are not many values here, and this is something that we have to explore. There are some other data that we have unpublished, and these were collected with MIMS. That's the MIMS that I have, which is available for the training course or lead experiment. If someone, we have a volunteer here to come to my lab, and we brought that to a cruise, and we ran a number of samples here, which were like time series, but we only collected the information from the first 24 hours, because after that there was a growing in the organism inside there. And mostly they are from the eastern basin of the Mediterranean Sea, from the Levantine and Ion Ionic Sea here, and someone close to the Alberine Sea also. And you can see that the values, the RETS values, are pretty similar. They're ranging from 0.3 to less than 0.05, about that. So the average or the median is 0.1. So this is the more or less close to the value that Christensen found for bacteria that they are in almost senescent phase. So this is like, let's say, a safe background value for respiration if you want to apply ETS. But this doesn't mean that all the respiration in all the regions in the ocean must have this relationship, because probably there will be more active bacteria in some areas. With applying this value of 0.1 of RETS, in also almost 20 years ago, we collect and we compile, I think it was the first estimate of dark ocean respiration in the ocean. And we collected all the ETS that we have there, and we plot all together. And you can see here that in general, there's an exponential decrease with respiration, but there is something, some peaks here in an area which is in the boundary between the central and the intermediate and deep waters here. And we don't know what is this caused, but it could be caused by layers of migrant, but could be caused also for difference in density where organic matter accumulates. We have seen that in some studies. So, but there is some variability here at these peaks here between 1,000 and 2,000 meters, more clearly up to the 1,000 meters here. And we made a very rough estimate, extrapolating this to the whole ocean, and we got a value of 1.2 petamoles of carbon per year, which is not far from what we would expect from all the carbon fluxes together there, and from the biochemical estimates also. So in fact, we compare here in this chapter of the book, where Jack is also co-author here, we compare in general terms the different approaches of looking at respiration by ETS with the oxygen utilization rates derived from apparent oxygen utilization and tracer from Jenkins and Wallace from the Sargasso Sea, and some other estimates. And there was a pretty good agreement in general between the ETS and the biochemical estimates here. There are also other There are also other studies that find a pretty good agreement between ETS and other geochemical estimates. And this is a paper by Vieito Fernandez, a very, very nice paper carried out north of the islands here in the surroundings of the stock station. He did here a tracer conservation model using climatological data from oxygen, DIC, and nitrate. And he estimated remineralization rates in the water column and compared with a data set that we had from several samplings at the stock station looking at ETS, and the correlation is really good. And also in this paper, if you have the opportunity of reading it, it's quite nice. I like it. He compares the differences between the stock and the batch station here in terms of carbon fluxes and remineralization rates. And one thing which is quite interesting is the high dependence that remineralization has on the suspended, particularly organic carbon, in this case of the stock, compared with the batch station. So sinking is higher in the batch station. So the production it's more about the same, but the sinking is higher here. But there is a lot of a lot of transport from the coastal dwelling into this area, and this contributes largely to the remineralization of the POC in the stock area here. And this is also this paper that Gerker showed the other day, the paper of Federico Baltar also, and it's one of the cruises of Gerard Kendall. And here also is clear dependence of ETS activity to POC and not to DOC along this section going through the North Atlantic ridge here. You can see there is a very, very good agreement, particularly here in the intermediate Antarctic water. There are some other more recent papers that they show also the variability, which is zonal variability, but also seasonal variability, not only in the POC and the DOC in an area like this one, where there is a lot of coastal offshore advection, but also in the ETS activity. So this corresponds to two cruises, which is Coca 1 is one cruise and Coca 2 was the second cruise, and two different times of the year. We also sampled two different sections, one north and one south here of the Cape Verde frontal zone. So here you have the two cruises here, Coca 1 and Coca 2, 26 north and 21 north. You can see that there are differences here. And you go to you look to multiple linear regression statistics, you see that most of the variance is explained of respiration is explained by the suspended POC, both in the epipelagic layer, but also in the mesopelagic layer. So here is a large dependence of POC on respiration, which is not always the case, because when you don't have so much exported organic matter from the coast, sometimes this DOC contributes more than POC to respiration. So this is something that you can infer also from ETS by this regression statistics between the different measurements that you take in the water column. And these are some, just to finalize, there's some data that we have still unpublished. And we have published some papers on this cruise, but not still this. This is a very, very nice cruise that we ran from the region of the Levantine water here in the Eastern Basin of the Mediterranean Sea. And we went through following the Levantine intermediate water, but also down to the bottom of the water column, exiting here and forming the, the Mediterranean, sorry, forming the Mediterranean water down to the Canaries here. And here you can see the ETS activity, the POC and the PON. You see that there is not, there's a quite good correlation between POC and ETS, which is basically driven by the larger values here, which are in the surface. But in the low values, I don't think there is a very good correlation here. And you can see, for instance, that the respiration here is low, but here you have more POC and PON than in the Eastern Basin. So this is something that we are starting to work on it, but I think it's quite challenging and quite interesting. So how we can compare and try to interpret why respiration is not higher where you have more POC, but it's similar or not like in the other places. But the resolution of the ETS gives you all this variability in a quite nice view. So you can look at these processes that with other approaches probably won't be possible to do this. In particular, this one here is the outflow of the Ionic Sea here, which is from the Adriatic Sea to the Ionic region here, and it goes down to deep in the water column. So one of the things we're starting to work on is to try to balance the production in these areas with the respiration in the different water zones here, water layers. This is wrong, sorry. This should be 200,000. So this is the mesopelagic, and this is the epipelagic and the bathypelagic. So we are using to convert our ETS of 0.1, which is the one that we got during the cruise, and we are using also from the relationship that I show you from the surface water here, and it has a lot of sense. I mean, there are some things that are very, very clear, and if you put all the values here and you compare with the prior production, it has also a lot of sense here. So just to summarize, on the way forward, The first thing I would say is that the enzymatic ETS technique, while it represents a fast and sensitive method to address this respiratory activity, which is not the actual respiration, at a spatial and temporal scale, which are very difficult to be addressed by other ecological approaches. So I think that potentially is a good method. At global and even regional scales, the ETS derived respiration, if we use a low respiration ETS ratio, seems to agree with other biochemical estimates inferring oxygen consumption in the mesopelagic zone. But we know that this ratio might vary in cultures of bacteria found in surface waters up to an order of magnitude. So we need to run experiments looking at changes in this respiration and ETS of mesopelagic bacteria cultures under replenished depleted organic matter sources. We need to understand the scale and cause of its variability in dark ocean, and we need to bracket this if it is possible, because otherwise we will be just playing with or assuming that all the bacteria are in the same physiological state in the deep ocean. So I think this is one of the main challenges that we need and we have in this future. But this is very, very difficult to do it on situ. I mean, measuring respiration in situ and looking at the bacteria in situ, basically because the rates are so low in the ocean that we cannot just go there and measure there in a very simple way. And finally, and this is one of the goals of the um, recent project that hopefully will be funded. Martin, Iris, and Giorgio, you are there. Is from the Ocean ICU. Is to explore the dependency of ETS-derived respiration on environmental variables in the mesopelagic zone, for instance, temperature, oxygen, organic matter, to derive global parameterization that could be implemented in biochemical model. And for part of this, we have applied for a ship time for 2024, 2025, for very long cruise going from the Canaries to Walvis Bay, crossing areas of very low oxygen, and changing temperature, different productivity, organic matter, and so on. So let's see, that would help to work in this. And that's all. Thank you very much. I don't know if I have been very fast or very slow, or thank you very much.
Just perfect. And thank you very much for this presentation.